Well, good morning, AP Bio students. Mrs. Shelton here, and I'm actually just going to give you a quick little lesson here on Chapter 3, Section 1, and Section 2. I've already given you Screencastify lectures on macromolecules, so make sure that you are referencing those when we get to section 3 of carbohydrates. There's a Screencastify already that talks about carbohydrates and lipids specifically, and then I also have a Screencastify for you for proteins and nucleic acids, so make sure you reference those. But the new information that I need to make sure that we're covering is the section 1, just talking about carbon and its unique properties and what makes that the basis of all organic molecules. And then also we need to talk in section 1, we need to make sure that we're understanding our functional groups as well. And then in section 2, we need to make sure that we are clearly understanding then our dehydration reactions and hydrolysis. So in this first picture that your presentation starts with, we do need to understand that this is a macromolecule. This large organic macromolecule, this is actually a protein that is being modeled here. And what you've observed already is that there's lots of different ways that we can model these large molecules. So with this is actually a ribbon type of model where we're seeing these alpha helices. We're actually seeing then some of these cofactors that are necessary in order to build the protein, the folding of the protein in order to give it its unique shape. But this is just one of many different organic compounds, and more specifically macromolecules, that we have to learn in Chapter 3. Now, first and foremost, we do need to identify that what we're seeing here, when we've got these carbon-hydrogen-based molecules, we are talking about organic compounds. So all organic molecules are always going to have carbon, and they're always going to have hydrogen in order to satisfy bonding rules. Now, carbon is very unique, and why carbon actually is this backbone of all organic molecules is that carbon can make 4 bonds. And with its 4 bonds, carbon has the ability to form long chains. It can form double bonds. But with this, we can also see that carbon can have branching patterns. It can have ring-shaped patterns. So carbon is extremely unique as the foundation of all of these molecules of living things. Now, we also have to make sure that we are understanding these 4 basic atoms. And with these 4 basic atoms, these are the ones that you're going to be addressing most often. Organic chemistry is no different than inorganic chemistry other than the complexity. But all bonding rules are still the same. It's still carbon has 4 electrons in its valence shell. It wants to make 4 bonds in order to complete its outer level. So it therefore has a valence of 4. Nitrogen, it has 5 valence electrons. It has a valence of 3 in order to make 3 covalent bonds in order to satisfy its octet. Oxygen has 6 valence electrons, but a valence of 2, meaning it wants to make 2 covalent bonds in order to achieve a full outermost level. And then hydrogen, remember hydrogen, this first energy level can only hold a maximum of 2 electrons. So there's 1 valence electron, but it also makes 1 bond. So we often see carbon being this unique 4-bond atom that now can make, again, long chains, branching patterns, etc. And then hydrogen then is often the atom that is used to satisfy any extra bonding that's there. Now, with macromolecules, we're going to see these atoms in our macromolecules. Macromolecules, though, we'll have to make sure that we're learning the monomers and the polymers of our 4 macromolecules of carbs, lipids, proteins, and nucleic acids. Now, what we are seeing here is actually classified as a hydrocarbon. Hydrocarbons are organic molecules made of only carbon and hydrogen. What we should know about hydrocarbons is that hydrocarbons are, these are non-polar covalently bonded molecules. So hydrocarbons in just this pure sense are actually not soluble in water. And where other molecules, when we talk about carbs and we talk about proteins and we talk about nucleic acids, those are soluble in water. When we do talk about macromolecules or we talk about organic chemistry, we do have to understand that there's lots of different ways that the same type of atom can be put together. For example, here I've got pentane and I've got 2-methylbutane. These, if I wrote out the chemical formula for these would be C5H12. This would also be C5H12. but their different arrangement gives them different chemical properties. So they have the same number and type of atoms forming the molecule, but their arrangement is different. So we classify those as structural isomers, is what they are. Now, this is the most common type of isomer that you will encounter are structural isomers. So what I don't want you to assume is that if you see C6H12O6, don't just assume that that's glucose. It might not be. So you need to make sure that you are recognizing this concept of what an isomer is. Now, also when it comes to a carbon-carbon double bond, functional groups can lie on different planes of this three-dimensional molecule. So this is where I can have cis-trans isomers where now functional groups end up, again, arranged differently about this carbon-carbon double bond. Now, some of our larger molecules can form enantiomers. And an enantiomer means that it ends up being a molecule. It's a mirror image of the other molecule. Now, what you want to look for in a possible molecule that can have an enantiomer is, do they have an asymmetric carbon? And what that means in an asymmetric carbon is it's a carbon that actually has 4 unique functional groups attached to it. So there's no symmetry to it whatsoever. It's asymmetric, meaning there's 4 different bonds that are completely unique. But those would be your different types of isomers that would all have the same chemical formula, but the arrangement is different with different chemical properties. And remember that even with molecules, form fits function. Now, functional groups, all I need you guys to know at this point in time is I just need you to recognize the functional group. When you see it on a molecule, you can recognize this OH as a hydroxyl group. You can recognize a carbon with a double bond to oxygen only as a carbonyl group. And so don't worry about the compound name right now. We do not need to worry about that. Right now, it's all about recognition. So my carbonyl group is going to be this carbon double bonded to oxygen. And now if I have a carbon that has a double bond to oxygen and it has a hydroxyl group, well, this is its Hollywood name of this Hollywood couple name. It's now your carboxyl group is what you end up having. This COOH is actually going to be very important when it comes to looking at fatty acids and also at amino acids and how they bond. Now your amino group, this is NH2. So this NH2, recognize it in 2 different forms. You can actually have it as the ionized form, where now it's gained a hydrogen, so it has a positive charge, or it doesn't have the hydrogen. But your amino group is going to be your only functional group that has a nitrogen, but this is NH2. This is going to be really important when we're recognizing amino acids. An amino acid has an amino group on one side, a carboxyl group on the other. Another functional group that's important in understanding protein structure is this sulfhydryl group. Your sulfhydryl group has a sulfur and a hydrogen on it. Now, why this is significant is there is an amino acid that has this functional group of sulfhydryl. So that's what we're seeing here. We're seeing an amino acid. And on this R group, this is the sulfhydryl. Now, sulfhydryls actually want to bind. They want to bond and form covalent bonds with other sulfhydryl groups in the polypeptide. So this is something that stabilizes then the tertiary structure of a polypeptide. Now, phosphates are also incredibly important for us to identify when it comes to our nucleic acids. Remember that a nucleotide consists of a phosphate, a pentose sugar, and a nitrogenous base. These phosphates are a PO4 2 negative, or you might see it spelled out as OPO3 2 negative. But this phosphate, being a 2 negative charge, also contributes to what we need to make sure that we're understanding. Oh, sorry, I don't have that picture on here. Is when we talk about ATP, ATP is essentially, it is an RNA. It's an RNA nucleotide with 3 phosphates attached to it. And those are high energy bonds in order to get these negative functional groups to now form covalent bonds with one another. Now, the last functional group I need you to be aware of is the methyl group, that's CH3. This particular functional group is going to become really important when we start talking about DNA methylation. We're trying to control our DNA and deactivate segments of our DNA in specialized cells. So functional groups, just be able to recognize them, and then we should be able to start recognizing them on the monomers as we start talking about macromolecules. The last topic that we need to address for 3.2 today is actually understanding dehydration reactions versus hydrolysis reactions. Now, in order to be able to understand this, we need to know the term monomer and polymer. Your monomer is the repeating unit of a large macromolecule. So for example, if we're talking about carbohydrates, we are talking about monosaccharides. Well, if I had a whole bunch of glucose molecules, those monosaccharides that are common in our blood sugar, and now I needed to link those up into something like glycogen. Well, the chemical process that happens is a dehydration reaction in order to link those monomers into large polymers. In order to do that, I have to remove a hydroxyl from one monomer, a hydrogen from the other, forming water, in order to now make these want to covalently bond to each other. So we call this a dehydration reaction because I'm removing water in order to join two monomers together. In this long polymer, if I am joining 4 monomers together, well, this also means that I pull the water out here, I pull the water out here, I pull the water out here. Everywhere that there's a covalent bond, there's been a water removed in order to now create this new covalent bond. Now, hydrolysis is actually the reverse. If I need to take that glycogen and break it down back into simple sugars, well, now the reaction is a hydrolysis reaction. Enzymes, hydrolytic enzymes, are going to break this bond, but also return a hydrogen and a hydroxyl to these monomers, so they're now stable monomers. Now, I just gave you examples of carbohydrates, but we need to understand that all 4 macromolecules, so whether or not I'm forming a triglyceride, between that glycerol and 3 fatty acids, it's going to be a dehydration reaction. If I'm forming long polypeptides, it's going to be a dehydration reaction. If I'm now forming a polynucleotide of DNA and RNA, it's going to be a dehydration reaction linking those nucleotides together. And if I need to break any of those down, hydrolysis that breaks those down. So these are terms that apply to any building of polymers or breaking a polymer back down into monomers. So for today, that is what I need you guys to take out of chapter 3. Section 1 is what I need you guys to make sure that you are understanding functional groups, isomers, and then also your dehydration reaction and your hydrolysis reaction. So